Hello, everyone. Welcome to this webinar. We're going to be starting in just a few more minutes. And today is actually the first session of the SARS-CoV-2 training program with LBRN. So I'm glad to see everyone here. All right. Hello, Mohit. I guess we should start. It's 10.
Yeah, hi Ilya. Hello everyone. Okay, sorry for the delay. I was just setting up the Zoom. Okay, hello everyone. How's everyone doing? Yeah, we will start in a minute. Let me share my screen. Ilya, can you see my screen? No.
No, not yet. Yeah, now we can see it. Yep, great.
Okay. So hello everyone. Welcome to today's session. Today we have the first session on the program for COVID-19, and in this program we will be discussing several aspects. And today in this session, I will start with discussing about the program in brief, and then we will learn about yourselves, like if you have gone through the registration process, if you have gone through the resources that we have been sharing. So today we will also discuss on the later part of this presentation the application of NGS data for the application of understanding the different aspects of COVID-19 genomic analysis. So just 1 second, my presentation seems to be stuck. Just a second. Sorry for this, guys.
Okay, so here, maybe I should then take it from here, Mohit. Yeah.
Sorry, Ilya, just some problem with the presentation. Is like I'll just take a couple of minutes. Maybe meanwhile we can help who have joined to go through the learn registration.
Okay. Okay, yeah, so let me share a couple of things. So welcome everyone. This session is the first session, so we're going to be going through some of the basic things. But before we do that, I'll let Mohit kind of figure out what's with the slides. Let us go through some of the major topics that this program is intended to address, and briefly talk about how you can get started with the program itself and get yourself into the content that we've prepared for the program. So first of all, the topic of this program is really SARS-CoV-2 as an extraction of a program that we have regularly called Bioinformatics for Infectious Diseases. So SARS-CoV-2 is an unusual virus in the sense that it has turned very quickly into a pandemic that has spread around the world, but also we are living at a time where for the first time in history, the data about the genomic characteristics of this evolving virus is being made available almost in real time. So if you're following the news and you don't live in a cave on Mars, you hear about this all the time. And what we know today is that the general public is being so aware of terms that just a few years ago, anyone would just ignore completely. What are mutations? What are variants of concern? What is the difference between the variant of concern and an actual mutation that has some pathogenic consequence? What are the different immune responses, right? So all of these topics are being discussed by the broad public. And it is important, we think, for us to focus on the aspect of bioinformatics in this program so that anyone that has some appreciation for the molecular biology and the pathology of this disease can actually base their opinions and their findings on data. And so that is really the objective of this program. Okay, so again, for those of you that are joining today, the first thing that we would like to do is we would like to today introduce you to how you can get started with learning about the different types of data available for SARS-CoV-2. How can you understand what this data really means? And how the interactive sessions that we are planning for this program can really help you become proficient at analyzing this data and asking important questions that are on everyone's mind. And so to get started, let us go to the OmicsLogic Learn portal. So let me ask all of you, how many of you already have registered on this portal? So this is the link I'm putting in the chat. And maybe you can let me know when you are registering, when you're in, you will see your email address right here. So please let me know how many of you have already registered. You can put it in chat, just put number 1 or yes, or something like this. So I'll give it a moment. All right, excellent. So if you have already registered, you can click on this email address and you should see your profile. So the first thing that we would like to do today is to make sure that your profile is completely set up. And I will show you how this profile can help you get into the program and start with the different material that we will be covering. Now, if you haven't registered yet, take a couple of minutes and register. So again, once you are registered, you should see your email address right here on the top. And by clicking on this, you will be able to see your profile with your profile image, social media links, a little bio that you can add about yourself. And then we will talk today about this progress tab. As you can see, the different activities that you perform on this portal are being tracked to collect information about you for your own benefit so that you can track your progress, but also because of the programs that we're going to be talking about today. Okay, so once you complete your profile, the next thing that we want to do is we want to go right here to the Programs tab and take a look at the program for SARS-CoV-2. Okay, and so here is the program page. If you're already a part of this program, you should see what I see on the screen. If you don't see this, you just see a bigger screen with the title of the program, and when it starts, you can see today is July 12th, so it's starting today. You need to still complete your registration. So it's okay if you haven't completed today, and up until the next session, you have plenty of time to complete the registration. But I want to start with a few key points about how this program is actually going to work. So the first thing that we will do is review the history and the pathogenesis, the origin and the pathogenesis of this novel coronavirus, so that we can get acquainted with some of the basics, with where the virus could have come from based on the available data. So what are some approaches to analyze this data and ask the questions about origins? How do we monitor the evolution of this virus to understand how it changes as it spreads? And then to also look at the responses to the SARS-CoV-2 virus to really understand what is the immune component. So how does this virus cause disease? How does that change between individuals? And also, how can we, based on our understanding of the immune responses, intervene? So what are some of the strategies for drug design, vaccine design? How do we think about these interventions in the context of what we can see from data? And so to facilitate that, you can see that down here we have a syllabus of the program where we will talk about how viral sequencing is being done, which is the topic of today's session, how we can find presence of viral genomes in different types of animals. As we know, pangolins, bats, even dogs have been suggested to be susceptible. How we can use existing databases like NCBI virus and GIS-AID to find sequences, how we can find high quality COVID-19 genomic data, and then how to start analyzing this data from the perspective of multiple sequence alignment, building a phylogenetic tree, then looking specifically for variants of concern so that we can track the alpha, beta, gamma, all of these different strains and understand how they evolve and what are the associations between different strains and different regions, and what are these variants of concern that we should be mindful of. Now, inside of this schedule, you will see links to the Zoom sessions. So how this session is organized is that we will review some of the content that will be also provided asynchronously. After completing the asynchronous content, you will be able to ask questions and interact with the mentor, and I will introduce you to the mentor today. And also, you will be able to run analysis together. So hands-on practical analysis that will be guided, and then you can look at the results and obviously check that the results are correct. So this is the schedule. Now, at the end of this, the idea of this program is to allow you to understand where to find data, understand how you can ask questions of this data, know how to perform analysis and plan your analysis according to the question that you have, using several examples from public repositories and peer-reviewed publications, and then potentially to use that to develop a project of your own. So at the end of this process, you will have a little bit of time to work on a project of your own, which you can start right from the very beginning as you learn about different aspects of this kind of analysis. Okay, so Maddie, does that answer your question? Okay, great. So now, as you go down below, okay, this is not loading because I'm sharing my screen. Let me just reload this. Okay, but included in this program are some asynchronous resources. And so to talk about the resources, I want to introduce Dr. Harpreet Kaur. She will be guiding some of the sessions and doing some practical interactive assignments together with you. And so I will give her the presentation so that she can talk a little bit about her background, but also to introduce some of the contents that you will have access to through the program. Dr. Harpreet?
Thank you, Ilya. Just let me share my slide. Hello, everyone. My name is Harpreet Kaur. I have a PhD in Bioinformatics. Currently, I'm curriculum developer and research consultant with Bioinformatics. In my current role, I am mentoring number of the research fellows and assisting participants from different programs to learn about data analysis, machine learning, visualization in the application of large data used in the biomedical research. So to provide hands-on training and to make participants familiar with approach to design and complete a research project, we are continuously designing different research projects using publicly available data and implementing various bioinformatics tools which are integrated on our T-Bioinfo server. So here we have collection of the research project where you can learn how bioinformatics can be applied in diverse fields using publicly available data to drive biological and clinical significance from the genomics data. So let me give you a glimpse of one of the project that is based on a current pandemic of the SARS-CoV-2, that is origin of the COVID-19. So as we know, whenever a pandemic outbreak happens, like the current COVID-19, so there are the several questions that needs to be answered. These questions can be of any of the following. For example, where did the novel coronavirus comes from? Which animal, what location, and how did it start infecting the humans? So to understand and design the vaccine against any virus, we need to find answer to these questions. So towards this, in the March In the March 2020, so Anderson et al. Have published a research letter in the journal Nature Medicine in which authors studied the proximal origin of the SARS-CoV-2 based on the NGS data, and they also find some of the important mutations in the spike protein that are responsible for its enhanced pathogenicity. So based on this study, we designed a research project. So in this project, we explored this pathogen coronavirus itself, as well as the disease caused by it. So here you will learn how you can find its origin and how much it is related with already existing human coronavirus strains based on various techniques like multiple sequence alignment and phylogenetic analysis using the publicly available NGS data. Next, you will learn what are the important components of the virus through which they enter into the human cell. We will also look into how much it is different from other coronavirus strains like SARS, MERS, etc. At last, you will also learn like how you can identify important signatures that can significantly different under therapeutic treatment or untreated conditions. As, as the genomic data continuously growing, so there are a number of the public repositories that maintain NGS data for the SARS-CoV-2. For example, NCBI collected all of the viral genomes from the pandemic and associated viral sequences of the SARS-like viruses in a new repository which is called NCBI virus. So during this program, we will learn how to obtain NGS data for the SARS-CoV-2 from these public repositories using specific keywords like its location, the host, as well as which countries it belongs. So after downloading the data from the NCBI, next you will learn how you can perform multiple sequence alignment to understand the phylogenetic relationship between these organisms. So here you can learn its genetic origin, and you will also learn how you can compare it with other existing strains based on the NGS data. So subsequently, you will learn how you can perform multiple sequence alignment to understand the mutational changes that are taking place in SARS-CoV-2 in comparison to the other strains from different animals as well as the other coronavirus strains from the human. So you can compare and find out some important mutation in a specific protein or a specific sequence. Next, you will also learn how you can analyze multiple sequence alignment to understand the differences and the mutation among different samples at nucleotide as well as the amino acid level. So you will learn how many mutations are synonymous and how many mutations are non-synonymous. So let me ask one question. Can anybody tell me the difference between synonymous mutations or non-synonymous mutations? So can anybody tell me the difference between synonymous and non-synonymous mutations? So you can post your answer in the chat box. Or let me give you just a hint. So silent mutation. Any one of you familiar with the silent mutations and missense mutations? Okay, so we have an answer from the Jahra. Phenomenal associated with the phenotype change. Perfect. So Maddie is perfectly all right. So synonymous mutation when they does not affect the amino acid, while non-synonymous mutations when there is a change into amino acid. Yes, perfect. So you will learn how many mutations are actually translated at amino acid level and how many mutations are remain synonymous mutation. So the next you will learn what are the important protein components in the virus as well as the host that are responsible for the pathogenicity. For instance, how ACE2 receptor binds to the spike protein, and you will learn how to visualize structure of the protein and its complex, that is ACE2 spike protein complex, using different publicly available softwares. So in the subsequent part, you will learn how you can obtain transcriptomics data for the patients or the host that are infected with the SARS-CoV-2 from the other public repository like GEO and SRE. Okay, let me ask one question. Can anybody tell me the difference between SRE and the GEO? So what kind of data we can find for SRE and what kind of data we can find from GEO? Anyone have some idea? Like what kind of data? Let me give you just hint. Geo, micro, array, SRA, NGS. So exactly not, maybe let me give you one hint. So one is more or less process data, other is raw sequencing read raw data. One of these repository mainly maintains process data and other repository mainly maintains raw data. So Dhruv is saying SRA maintains raw data. Okay, yes, perfect. And yes, Manoj, you can ask your question, please. So you can post your query in the chat box or otherwise you can unmute yourself. Okay, so Manoj Kumar Jhana, you can post your, can you enable? Okay, so Ilya, can you just enable or just so that he's able to unmute himself? Or you can unmute yourself, I guess.
No, I mean, I think as a co-host, you can probably unmute him if you
Oh,
need to, or you can just, yeah, put the answer in the chat.
Hello, ma'am.
yes.
Ma'am, good evening. Thank you so much. Ma'am, I have a question. Now I am a second year in a biotechnology subject undergraduate student.
Mm-hmm.
Ma'am, I want to pursue in future biotechnology in MSc or bioinformatics. Which is the better, you know?
So that depends upon your research mainly interest. So I think according to your research interest or what is your goal of, I mean, so accordingly you can proceed. Otherwise, uh, bioinformatics and biotech are I think both are equally good.
Both are equally good. But ma'am, which one is better for me? I don't understand. That's why I raise my hand.
Sorry, Dr. Preet, to interrupt here. So, Manoj,
Oh,
maybe we can answer this later because it's more relevant to your work. So, maybe we can do this later once the presentation is over. Thank you.
yes, we can. I think at the end of the session we will address those questions. Thank you, Manoj. So here you will learn how to explore this transcriptomics data from the host to find out important signatures that are associated with the drug treatment like ruxolitinib. Similarly, you can analyze such data to understand how virus cause infection and what are the some of the immune response differences between acute or the mild COVID-19 disease condition. So here you will learn how we can perform phylogenetic analysis. And let me demonstrate to you how we can use our T-Banfo server to perform phylogenetic analysis using NGS data. So let me give you a simple demonstration. Okay, so this is the main analytical platform. And here you will find different sections for different types of analysis depending upon your goal, depending upon your data. So here under the data integration and the modeling section, you will find this phylogen under the data association subsection, you will find this module phylogenetic tree. So here, let me just give you this project that is demo SARS-CoV-2 origin. So this is an example project where we have already provided the data. So once you're able to click on that, then you will direct it to this interface. So while clicking on the start, you will able to upload the data. So here you will learn what kind of data is required. For example, what should be the format. So like here we have the NGS data in the FASTA format. Then how to upload the data. So while clicking on, we will able to upload this data because this is already example project. So here we need not to upload data externally. So we had already uploaded data here for you. Then the next option, which is automatically get highlighted after uploading your data, that is multiple sequence alignment. So it will align your sequence, all the sequences using cluster W tool. Then once you have at a sequence alignment, next is their alignment at the amino acid label. So here the first, because in this example project, we have also uploaded reference genome with translation codons. So while clicking on OK, then we will able to perform multiple sequence alignment at amino acid label. So then we can select a specific codon position. So eventually you have different methods to generate phylogenetic tree. So here, for example, we have AIP method, then we have based coalescent constant size, based speciation Yule process, with speciation birth death process. So here we are using right now all the methods. So we are selecting this option, all phylogeny method options. Then by clicking on OK, we will able to perform to generate phylogenetic tree with all the methods. So finally, we will get this option, tree space. So this is another module which will generate a median tree for you. So while clicking on this, we able to generate median tree. Then finally, by clicking on the end, we will able to complete our pipeline. So then once your pipeline get completed, then under the download output file, so you will get various types of output. Now, let me just show you one of the output here. If I go to the presentation again, so this is the output file that we will obtain. So this kind of phylogenetic tree you will get. So here you can find out like you have sequences from different host. For example, here we have human SARS-CoV-2, pangolin coronavirus, vet coronavirus, vet SARS, and vet coronavirus. So then to understand what is the phylogenetic or evolutionary relationship, and for example, how this human SARS-CoV-2 related with other coronavirus strain. So from the based on the phylogenetic tree, we can understand that it is closely associated with the pangolin coronavirus.
Okay, Harpreet, let me interrupt you for a second. I see a few questions that maybe we can address briefly. I think
Sure.
that they are relevant. So number one, for those of you that have completed the registration to the program, you will find the link to the Slack channel at the bottom of the program page. So if you're not a part of the program, that's why you're not on the Slack channel. If you're already a part of the program, then please find it at the bottom of the page. Again, I can just demonstrate where that would be. Just 1 second, let me share my screen so that, you know, as you're following along. Okay, so here's the page. If you scroll all the way down, right here, if you click on this, ask question, you will be taken to the Slack channel. And then it will open right here in Slack. Okay, so if you, Livia, if you have completed the registration, are you logged in? Can you see your login information? Okay, maybe you can share your profile ID and I will take a look. So if you click right here, this whole URL, that is what I need. Okay, anyone else that also has completed the registration or has registered through LBRN and you don't have access to something, just post your link right here and we will check during today's session. Now, the other question that we had here, can you use the Bioinfo platform to access files saved on GCP, Google Cloud, or AWS, Amazon Web Services clouds? Yes, any cloud-based link that you can share with an FTP or HTTPS, you can actually you can share that. Okay, so Olivia, it looks like you're a part of this program. So if you go to your page, oh, okay, so I think this is the older one. So let me take a look at this and just double check your account. Okay, for any questions about publicly stored data, yes, you can use it using HTTPS links or FTP links. If you want to download SRA files from NCBI, you can use that directly as well. We will go through that in the program sessions. Okay, so back to you, Dr. Harpreet. I will check her account now.
Thank you, Ilya. Okay, so for example, this phylogenetic tree shows that human SARS-CoV-2 genome sequences are more closer to the pangolin than to wet coronavirus. So that's depending upon the data that we have used in this project. So if you're interested in getting started with such analysis on your own and already you are registered, so I recommend you to going on the Learn platform and how you should start different courses. Now let's go over Learn platform. So the first lesson I would recommend you if you are very much new to the biology and the bioinformatics, first course that you must go through, that is Whites in the Molecule. Now let me share its link with you. So if you are already registered, you will able to access these lessons. If you have yet to register, then please go ahead with your registration and then you can start these courses. So this is the first course that I would recommend you should start. And then once you complete this course, then move to the next course, that is Introduction to the Bioinformatics. So let me share its link with you. So especially this course is for those who are not from the bioinformatics background, so that you understand what are the important applications of the bioinformatics, and we will also learn what's your background. So here you need to submit one of the forms. So based on which we learn what's your background so that we can accordingly guide you. So the next course, once you have completed these two introductory courses, then to understand how you can do different genomics data analysis once you have the NGS data. So the next course that I would recommend you, that is Genomics. So here you will learn the basics as well as the advanced of the genomics. So you will understand how to find data, the genomics, as well as what types of analysis you can perform for the genomics data. So once you have completed this course, then I would recommend you this particular project that we already talked about. So that is project COVID-19 origin and pathogenesis of SARS-CoV-2. So these are the courses you must start, like first complete white cell molecules, then introduction to the bioinformatics, then complete genomics, and then go to the project COVID-19, where you can learn how you can do different types of analysis once you have NGS data and how to interpret those results. And once you are doing all these courses, and please complete all the assignments. So most of the assignment and the analysis that you are going to run, that is here. So after learning or performing multiple sequence alignment, in the next part of the project, we will move to the variant data analysis. So then you will learn how you can create reports on the genomics sequencing data using custom R scripts. So then under the genomics course, you will find some of the important scripts where you can learn how you can create reports for the different variants of interest. So this kind of report you will able to get and of running. So once you have the data and GIS data, then you can find what are the important variants. For example, here number of the variants that found in the spike protein are the 3, and where are the different mutations. So then in each sample, what are the important mutation for each type of protein? Subsequently, you will able to create this kind of plot. So in each sample, at which position a particular nucleotides get mutated, and apart from that, you will also get whether this mutation is synonymous or non-synonymous. So in the next session, we are going to start with multiple sequence alignment and how to find data from the public repository. So now I would like pass this session to Dr. Mohit Mazumdar to talk about the upcoming program for this complete course. So over to you, Dr. Mohit.
Yeah, thank you, Dr. Preet. And I kind of shared with Maddie here, he having a question. So I think that's clear. Okay, great. So what I kind of wanted to start with was the program, but it has been so much described in such a detailed manner and a great manner by Ilya and Harpreet, Dr. Harpreet. So let me take you through the sessions, and maybe like very briefly, I can tell about the second phase of the program, which includes the genomic surveillance. So the first part of the program was, I think, is very well described, and you got to know about the techniques and the platforms that we will be using in the program. And yes, you don't have to finish all the coursework right now, but I mean, it's up to you how much of that you can cover till the next session. But it would be nice if you can finish the introductory courses so that we can start with the basics that we are planning to cover for the entire program. So we will be starting from the very basic of genomic genomic technology, and then take you through this different scripting and reporting and all of that. So yeah, again, let me try share my screen. 1 second, please.
Yes, and please, as Mohit is sharing his screen, if anyone has any questions, feel free to put them into the chat box, and we will try to address those questions today. So was it clear for everyone what we are going to be starting with in terms of the program? What would be the objective for the next session? I hope that Mohit actually answered that pretty clearly. Yes, Mohit, we can see your screen now.
Right, thank you. So as I was saying, that this entire program, and I think many of the students and faculty, the beginners who have joined, might have this question: is that what is this program? Who is this program for? So this program, this OmicsLogic program, is designed for students. It's designed for faculty. It's designed for beginners. It's designed for biologists, also for clinicians and researchers who are interested in advancing their understanding of the logic that is behind the analysis of SARS-CoV-2 genomic analysis, and also to understand the analysis of variant of concern, and then to develop a workflow of variant reporting using using curated data, as we heard from Dr. Harpreet and Ilya also about different repositories that are there. So these repositories have clinical data, environmental sequencing data. So this all will be used in the program. Again, this program will not require you to have any background in coding or in bioinformatics or in biostatistics, because we will be covering many of the topics, and most of the materials that we have prepared for the program will be available to you to go through. So you can learn what we are covering, and you can understand how to do the analysis yourself. So, I mean, what is the basic need to go through this program? Yes, I mean, it would be great if you know the topics related to cell and molecular biology, because those are the terminologies that comes often when we are doing these sort of specialization sessions, and involving biostatistics and also other scientific domains, right? And so this about the specialization, I think this program is a specialization for bioinformatics for infectious diseases. In that program, we cover different aspects of virology. But in this program, which is more specialized for SARS-CoV-2, we are going to understand we have few objectives, and that would be very much cleared. I wanted it to be very much cleared in the first session, because we are meeting for the first time and we all are getting introduced. So in this program, we together will be working on data analysis tools to help detect viral genomes using NGS data, and then viral strains, haplotypes, if you have heard about them. And then Dr. Harpreet did talk about synonymous and non-synonymous mutations. So we will be understanding this from the perspective of file types. So what are the NGS file types, like FASTQ, you might have heard of, or FASTA, that you surely have heard of, I guess. And then applying these into a comparative analysis and understand how does this matter, and how does this information is critical to do vaccine design, or to look for the principles of antiviral drug design, as well as to develop understanding of resistance from the standard drugs, which includes also like antibody resistance, right? So we have three mentors which today have presented, and you are free to reach out to us for any questions. So Dr. Harpreet is there, myself will be available, and Ilya is also available. So we will also put the email address that you can reach out to for if you have any questions, or if you're facing any issues. We also have a support team with interns and our community manager. So please feel free to talk about and discuss all of that. So in the next session, which will be about detecting viral genomes, and then from animal and patient samples. So in this session, we will be using NGS data to find the pathogen. So we will be using obviously bioinformatics pipelines and processing steps to structure genomic data. And then you will be learning about some of the algorithms, that are used to align these big data sets. You will learn about algorithms such as, if you have heard of Bowtie, it's a popular algorithm for NGS analysis, NGS data analysis, HiSat, STAR. And then we will be working about on identification of sequences using NCBI virus database, and then extracting reads information from alignments, multiple sequence alignments that Dr. Harpreet talked about. So this will be about the second session. So let me quickly take you to all of the sessions that will be covered in the program, so that you have a clear picture of how this is going to be. So the third session will be about extracting reads that did not map to the host genome from FASTQ files after alignment, visualizing how they align to a specific genome from a database of viral genomes. So there are databases that we'll be exploring, which will be helpful for us to understanding of genomic variations in short reads. After that, the first part of this program, it will be to extract data and understand how data is deposited and how we can utilize that publicly available data, because data is being generated every day, and huge amount of data is being generated for, especially when we talk about COVID-19, in this COVID-19 pandemic. So in this session, we will be finding the right genomic sequences. We will be using NCBI alignment to check the quality, and preparing data for analysis. So this will be the session after that, which is the last session on the second phase, for the first phase, where we will be going through step-by-step overview of a pipeline that is used to align sequence, translate trinucleotide segments into amino acids, and then use the alignment for phylogenetic analysis using these. So we also have example pipelines that we have created for you on the Bioinfo platform, and using that, you will be able to go through the example that we have put together comparing different coronavirus or sequences. So that would be a project that we will be covering in this session. So now the second phase of the program, which is more focused on genomic surveillance of SARS-CoV-2, and very much into the research of variants, and the effect of variants and variants of concern. What are the different types of variants? So in this second phase, we will be learning about, again, the relationships. So the relationships which we can understand from phylogenetic analysis, using different sort of algorithms. We will understand the relationships by strain, by population, and by host. So there's a lot of data available for each of these studies to be conducted. And then analysis of variants. So how variants could be analyzed once you have identified that this is a variant of concern, what are the functional implications, what are the structural implications, and the association of structural analysis to understand the function. So how can you correlate function structure with function? And then, I mean, to give you a brief understanding of how this is going to be, so the pre-processing or the processing of these data sets would require high throughput workflows such as these. So I discussed with you the algorithms, some of the algorithms that we will be using. So in these workflows, bioinformatics workflows, it is essential to, which are essential for this sort of analysis. We will be also utilizing the R package. So R statistical language that has a rich library of tools and functions that are available at Bioconductor, and also GitHub will be utilized in this program. So once we have the analysis, how do you go to from analysis to pre-processing and understanding that significance of that analysis? So all these relationships and what data has been generated, I mean, we have a clear understanding of what bioinformatics methods are capable of after working for so long on different types of data that has been deposited all around the world. So bioinformatics methods could help us find individual mutations. Then we can do sequence alignment that could combine with evolutionary models that can track the changes in the genomic sequences and then link them to a process. So these models, they fit to thousands of genomes, and they reveal this dynamic tree of the pandemic, which shows the virus that continues to change continues to change into different clades that separates it from the common origin. And that has been seen in this, like this is a free resource that we'll be covering in the session that you'll get to see. So yeah, so this will be also understanding of how different mutations are originating from different geographic locations. So as I was telling you that we would be also discussing few of the case studies. So wastewater sequencing is one of the projects. So in this project, we will be utilizing pipelines for processing. And then what happened in this project is that our team was able to study in this project about the genomic variations in the samples that we collected and identified mutations. So since the data was collected over time in this project over several months, this analysis revealed important information about viral transmission among students in comparison to the rest of the city. So it's a project driven by LSU. And primary results really encouraged us to seek for more funding to work on this. So to identify the novel strain that are emerging that could actually help the whole city and benefit. And so it's a genomic surveillance that is being utilized. So this will allow, I mean, local and regional governments to develop mitigation strategies that have been used to show that has been shown to be successfully implemented at LSU based on the PCR testing. So this is one of the case study that we will be covering. And then, as I was telling you, and Dr. Harpreet did showed us the tutorial, and we have prepared also tutorial and resources for the participants to guide them to create a report to analyze variants using genomic sequencing data, and then cloud processing, and then custom R script. So the second phase, this is designed into several sessions. So I get that many of us get overwhelmed with all of this information, but they are described in sessions that will be happening bi-weekly. So you will get ample enough amount of time to go through the material that is being shared, the live sessions, and the recordings will be also shared for you to review them at your own time, at your own pace. So as I was saying that we will be covering them through different sessions. So this will include hands-on sessions where we'll be working on clinical and environmental sequencing. And to understand this, like the project that you are thinking about. So different type of data sets, as I said earlier also, the different types of data sets from patients have been collected and categorized based on age, based on pre-existing conditions, social habits, lifestyle, climate, location. So there are several of these factors that are involved, social economical background when it comes to COVID-19. So I think those are the factors that are considering. And then about correlating the structure, the sequence, and the evolutionary analysis, and then mutations and the conservations and the viral evolution, modeling the relationship between the sequence based on probabilities. So we will be talking about phylogenetic analysis, evolutionary analysis, and conservation. So it is very important in the context of understanding the behavior of the virus, or to understand about the variations, or what are the guiding factors behind them. And then after learning about all of this, obviously we discussed this over and over, that the scripting that is involved to generate a report. But after that, I mean, sorry for that. Sorry about that. So after that, we will be talking about the interpretation of molecular level effect of these missense variations or variants, which is indeed challenging and requires an investigation at an amino acid substitution level. So we will be talking about the protein structure, which is really fascinating. We'll be learning about the 3D dimensional. How do we look at the 3-dimensional structure? How do we identify those mutations in the 3-dimensional structure? So what kind of questions that we are going to answer? So we are going to answer questions like: is a variant changing a site involved in key molecular interactions that has a key correlation with the change in the function? For example, I mean, if a mutation is at a binding site, or is at a place which is critically involved in interaction, I mean, what would be the effect? So is a variant changing an amino acid located at a protein core, or is a part of a cluster known to have pathogenic mutations when you see them in 3-dimension model? So these are some of the questions that are really interesting to learn from. And from from genomic analysis, if you can trace back to the structure, that is a very satisfactory analysis, because you're understanding from a very basic, from a very sequence level understanding, to you're reaching to a level where you're going for a structural level analysis, and you're correlating it with the function, and then you're giving a clear story. So, I mean, the future expectations, we will discuss that as well. So, in that, I mean, we will discuss how this different countries are using this in and investing billions of dollars in sequencing, variant detection, vaccination. So, what could be the strategies based on this for SARS-CoV-2? And about that, I mean, there has been a lot of news, and some of these old news that you see is that all of the governments. So in early February, US laboratories were only sequencing about 8,000 COVID-19 strains per week. And since then, the rate of sequencing has increased substantially. I mean, strengthening the country's ability to detect and respond to the emerging and more contagious COVID-19 strains, like the variants currently sweeping the Midwest and the part of East Coast. So to do that, like the Biden administration have already made a nearly about 200 million investment to help increase the genomic sequencing from 8,000 to 29,000 sample per week. And thanks to the funding from American Rescue Plan, and then CDC is involved. So we would get an understanding of increased geographical coverage of sequencing to better understand these and detect these emerging threats like variants. So, I mean, this means that both existing and new COVID variants could be detected faster before they grow prevalent. So, this is some of this and the application of this sort of analysis, and understanding the importance and the skill sets that are involved in learning this. So, I think it holds a lot of importance in today's world. And now, I mean, this is what I wanted to tell you all. And before I kind of wanted to answer a few questions that are out there. One of the questions was about career, about like opportunities, about how do we go about to the next step after learning this. So I would want to take you to this LinkedIn group that we have, where we keep on posting about job opportunities, about internship opportunities, about industry related opportunities where you can like go and work, because we have a lot of participants, a lot of collaborators from industry who would look for specific skill sets related to these sort understanding of detailed understanding of maybe genetic data, maybe clinical data, and all of that. So I usually get a lot of queries from our collaborators who are maybe CEOs, and all of them would ask that if you have a student who would be interested in joining in some of our projects. So having a good profile and then joining some of these groups would give you that information what is out there. So maybe I would request Celia or anyone else, or maybe Ojasvi, to share the link of the LinkedIn group. And similarly, I mean, as I was telling you about the LinkedIn group, similarly, if you're on Facebook, we have a group on Facebook as well, where we keep on posting about the upcoming programs, and you got to learn about what's going on and who's joining and who's what, and what are the opportunities that are there, and what are the projects that are ongoing. So this is an important aspect, I mean, that is there with this training, is to be able to collaborate with other people. I mean, we have a very rich bioinformatics research community, and that actually helps us to collaborate with a variety of researchers who are working on different areas of research. So if you have a research interest, and maybe if it is being shared by someone else, it's always great to work on a group project. You get results pretty quickly. You have diverse mindsets that can come up with different ideas. So those are some of the opportunities that I would want to encourage all of you to take in the coming days. Once your training is like more or less, you're more confident about all of the resources and have some experience going through the example projects and the courses that we have put together. So I think that's all what I wanted to tell all of you about this program, and I'll be happy to address any questions that you have. So you can see me now. Hello.
Thank you, Mohit. Yes, so this was
Yeah.
a good overview of upcoming sessions and how all of you can join. A few questions that I see. The first question was, can we also predict what kind of genetic modification might be taken by the virus based on the various environmental factors or any other external factor? So briefly, to answer that question, it's very important to know what is in the data, right? So this program is going to be focused on analysis of available data. If the data about other factors like location, age, environmental factors, etc., is available, then in the program you will be able to identify methods to find some correlation. Now, as you can imagine, association does not always equal causality. So while we have some data, there's also probably a lot of data we don't have. And so that is really the challenge in a lot of these bioinformatics projects. What is the available data? So if you're a part of the program, Purabi, you'll be able to share the data sets and even find the data sets in repositories that we will showcase. And if that data is available, then we will be able to discuss that in detail. The second question was about certification. So maybe Mohit, you could address that.
Okay, let me check that. I missed that, sorry.
Okay, so about certification, yep, you see Arup Ratan Biswas is asking.
Okay, yeah, got it. So in this program, I mean, there's another opportunity to get the certification by completing the course. So when you go through and finish Introduction to Bioinformatics, you get a course certification. And by the end of the program, we will give you an opportunity to discuss and submit your research proposal, as well as like if you participate and finish all of the sessions, you get a certificate of participation. And once you submit your proposal, there's an opportunity to get a certification of excellence, which we will be sending out otherwise. But you will get your certifications once you complete the sessions and earn points by completing the task. So if once you complete each of the task and the assignments, you get point for doing that. You get points to complete the sessions, also to interact and to do the interactive, to be a part of the interactive session. And you can complete that like once you, like today's session is done. So you can do that by clicking the complete button. So in that sense, you get points, and also like you kind of let us know that you have gone through this session, have completed the session. Other than that, I mean, there are several courses that gives you certification. So you will have an opportunity to earn a lot of certifications using this one single program. That's what I'm coming at. So there are opportunities to get certifications from different courses, and then there's this certification for the program that you are part of.
Exactly. So here under your profile, you will find all of your certificates. So you can see program certificates or course certificates. The way to get a program certificate is on the program page to complete the session. So today we had our first session. If you're a part of the program, you can click on complete session, and then you will see session completed. That adds towards your certificate. So now you can see down at the bottom, 1 out of 10 sessions complete. So once you complete all of the 10 sessions, then you will be able to get your program certificate. In the program, we have a number of different courses included, and each one of these courses comes with its own certificate. If you complete that, you will have a certificate for that course. Okay, so the other one, how to obtain the reference sequence or amino acid sequence for a variant like alpha or beta of SARS-CoV-2, as it is not available in NCBI for analysis studies. So that's an excellent point. Variants of concern are being tracked by multiple groups. One of those groups is a consortium called Spheres. Another one is a consortium called Pangolin. So we will take a look at exactly the mutations. It's actually combinations of mutations that are being tracked. And in our second part of the program, we will actually run analysis from raw sequencing reads to identification of variants of concern, and then building a report with all of the important variants that are listed on those websites, which is publicly being reviewed by the consortium and then posted. And so we will guide you on how to do that. So Sasta Kumar, let me know if that answers your question. Maddie is asking, I had some logistics question, such as how long will each Zoom session be? So the Zoom sessions are 1 hour, so we're a little bit over time today. Is there a place where we can tell that asynchronous coursework we should have done by each live session? So in the schedule, you will find information on what topic will be covered. And in the session itself, we will discuss what are the specific courses that you can get started with. Now, we know that many of you are coming with different level of bioinformatics already, understanding of molecular biology, understanding of SARS-CoV-2 sequencing approaches. So that is something that we are doing today in today's session. Maybe we can pause here and actually ask everyone to share a little bit about yourselves. And I will tell you what is the best way to share that information with us. So as was already mentioned by Dr. Harpreet, we have this course called Introduction to Bioinformatics. And in this course, why we ask everybody to complete this course is because one of the first things that you get to do is to fill out a form about your background and current occupation, as well as location and things like that, and what you are interested in. So if everyone could take a look at this course and submit this form, we will analyze the results from this form and appropriately to your level of background, recommend some coursework that you can finish so that by next time you are ready. But in general, we do recommend finishing Introduction to Bioinformatics, Bytes and Molecules, and start looking at this course called SARS-CoV-2. You obviously don't have to finish. If you do at least one of these courses, that will be great by next time. So please let me know, Maddie, if that answers your question. Okay, Livia, you have a TBioInfo demo user access. It will be enough for the program assignments. you will get access to the educational version of the license by next time. So everyone who has joined today, this session, after today's session, we will review everybody's accounts because the registration is still open. So you will have time to complete the registration by our next session. And we ask that if you are not registered yet, that you actually complete that registration, and that will give us the ability to issue different levels of account access. So we will have our next session on July 16th. Okay. Okay, are there any other questions? All right, excellent. So maybe I could ask from all of you that are present here today, if you have gotten access to the Slack channel, please let me know. If you have any technical questions, that would be the best way to interact with us, is post your questions on the Slack channel, and we will review those so that we can address any questions that you have. Apart from that, you can always reach out to marketing@pine.bio, Mohit@pine.bio, support@pine.bio, and Ilya@pine.bio. Please feel free to reach out to us, and we look forward to interacting with you, helping you along the way with this program, and we're excited to get started. Thank you.
Thank you, everyone. Have a nice week.